Protein synthesis starts with a promoter. Promoters are signals in the DNA molecule that mark some very specific bases or a promoter site that is needed for the making of proteins. DNA is a nucleic acid that is made of tons of nucleotides. Each of the DNA nucleotide is composed of 3 parts: a phosphate group, a 5-carbon sugar called deoxyribose, and a nitrogenous base. They can either be adenine, thymine, cytosine, or guanine. The phosphate, sugar, and base are held together by covalent bonds. And now that's done, let's start the transcription. So, the first part of protein synthesis, transcription, begins with the promoter signifying the RNA polymerase, which is an enzyme where it should begin making the mRNA. One thing to note is that the 2 DNA strands are held together by hydrogen bonds between the 2 bases. And why hydrogen bonds? It's because they are weaker. They can easily be broken by the RNA polymerase, which is what's gonna happen soon, during transcription. The RNA polymerase then breaks apart the hydrogen bonds between the 2 bases, splitting the 2 strands apart. Once it did that, we'll use 1 strand of the DNA as a template, which just happened to be this top one, and one by one, it will start bonding RNA nucleotide to the DNA strand. The addition of the RNA nucleotides follows the base pairing rule, except instead of matching adenine with thymine, it bonds with uracil instead, so as shown here. And finally, A to the last T. And also, the RNA polymerase would also help bonding the new strand of mRNA nucleotides together with the original DNA strand that is being read. Then, the mRNA would break away from the DNA and start traveling out the nucleus into the cytoplasm. Then, the 2 strands of DNA will join back together, ending transcription. That was a lot of digging, isn't it? Very similar to DNA. RNA is also a nucleic acid. Each of the RNA nucleotide is composed of a phosphate group, just like DNA nucleotides, a 5-carbon sugar called ribose, and a nitrogenous base that's either adenine, uracil, a new one that's not on DNA, cytosine, or guanine. And with that out of the way, let's begin translation. I'm sure that all of you are super excited at this point. After the mRNA exits the nucleus and enters the cytoplasm, translation begins. Ribosomes are cell organelles that are sites of protein synthesis that are made of 2 subunits, 1 and 2. And the mRNA would enter a ribosome and begin the assembly of the polypeptide chain. Codons are 3 specific bases on the mRNA that would code for 1 amino acid. For example, the codon AUG is a start codon, which means it would always be the first codon read. AUG code for methionine. As the ribosome reads each codon of the mRNA, it directs tRNA to bring the amino acid into the ribosome. The tRNA have specific anticodons that are nitrogenous bases that are complementary with the basis of the codons of the mRNA. So now I'm going to demonstrate how this works. So the tRNA with an anticodon of UAC comes in that bonds with AUG, which means the amino acid that it brings in would be methionine. And once the anticodon on the tRNA matches with the codons on the mRNA, the amino acid would detach from the tRNA and wait for the next tRNA to come in to bring another amino acid so those 2 can bond together. Once this matches up and it confirms, it will again just move away. And now there's 2 amino acids, and I will talk about what happens to these 2 later. It's important to note that there are only 2 binding sites in a ribosome, which means only a maximum of 2 tRNA can be in the ribosome at the same time. The tRNA needs to constantly move out to let new tRNA in. And the last tRNA comes in, and oh wait, what's this? A stop codon? That tRNA would not bring any amino acid in. What happens with these 2 amino acids? I'm gonna show you right now. The rRNA. What's the function of the rRNA, you ask? After a second amino acid is released from the tRNA, a peptide bond, the bond between the first amino acid and the second amino acid, would form between the 2 of them. After multiple amino acids bond together, well, in this instance, there's only 2, a polypeptide chain is formed. During the formation of the peptide bond, rRNA, these little things inside the ribosome, help hold the amino acid in place and carry out chemical reactions to join them together. And as I said before, the mRNA will stop being read once it reaches a stop codon. And again, the tRNA for the stop codon will not have any amino acid attached, telling the ribosome that the polypeptide chain is done. And finally, the polypeptide chain is released into the cytoplasm from the ribosome to be used as a protein molecule to perform their functions. And that is it. Thank you for watching.